Hello and welcome back. Today is all about cell division. We're going to talk about the three parts of cell division. We're going to talk about the stages of mitosis. We're going to talk about all the pieces that go into how cells divide. So to start off, let's talk about just mitosis as a kind of give ourselves a general working definition and some of the main reasons why we go through mitosis. So our definition of mitosis is going to be the division of the cell nucleus into two genetically identical daughter nuclei. Mitosis creates identical cells. Mitosis is involved in dividing up chromosomes so that each new cell has an equal number. This is how we make identical cells. And the daughter cells produced are diploid, meaning that they have two sets of chromosomes. The reasons that we have our cells go through mitosis, there's a number of reasons, but the four main reasons are for growth. When multicellular organisms increase size, they also have to increase the amount of cells that they have in their body. So to grow, you need to go through mitosis to create new cells. Asexual reproduction is done by mitosis. To repair tissues, you replace damaged or dead cells by creating new cells to replace them, and then during embryonic development. So a fertilized egg will undergo mitosis and differentiate into all of the different parts that will then make up the embryo. Now there are three parts of the cell cycle, and the first is the longest. So looking at this little chart up here, interphase, this green eye, includes G1, S, and G2 phases. So like 85 to 90% of the time that a cell exists, it is in interphase. This is the longest phase, and this is when the cell grows and the chromosomes replicate. So in the G1 phase, it's just the growth of the cell, the organelles are doubling, protein synthesis is occurring. In the S phase, that stands for synthesis, the G stands for growth, so growth 1 phase. The S phase is when DNA is actually being duplicated. This is when DNA replication is occurring. And then in the G2 phase, that's when we check for errors in chromosomes. So as DNA replication is happening, sometimes there can be a mutation there. And if there is a mutation, hopefully that will get found during the checkpoint stage in G2. If it does get found and there is a mistake and the cell recognizes that there is a mistake, the cell will stop and will not go through cell replication, or cell division, sorry. The second part of the cell cycle is mitosis. That is this like light orange, right, the PMAT. So all of that, those four parts make up mitosis. That is the actual division of the nucleus and the movement of chromosomes into two new identical cells. Then the third part of the cell cycle is cytokinesis. That's that purple sliver, really tiny, not a ton of time spent in cytokinesis. And that is just the division of cytoplasm, which is the like official formation of two new identical diploid daughter cells. Now, how does this all happen? Well, you can thank cyclins for that. They are proteins that control the cell cycle to ensure cells divide when new cells are needed, but not at other times, right? Remember I said that cells spend most of their time in interphase, so they don't need to divide 24/7. They do divide frequently, but they don't need to divide like immediately all the time, never stopping, because then you're just overrun with cells when you don't need that many. So cyclins are there to determine when to start each phase, when to move into another phase, and you see these like peaks of different cyclins, so you know which one is actually doing its job at the time. And how do they do their jobs? Well, they bind to enzymes called cyclin-dependent kinases. Those kinases become active and attach phosphates to other proteins in the cell. And once those proteins are activated, then they start to carry out specific tasks within the cell. So when cyclin E hits this peak, then we're going to move into our next phase, right? When cyclin A hits its peak, then we're going to move into, we're going to start this other process, right? We're starting our checkpoints, right? When cyclin B peaks, then we're gonna, okay, now we're going into division, right? So these cyclins have specific jobs and they're looking for specific enzymes so that they can start the process. Okay, so when we talk about cell division, we're talking about normal cell division, right? Mitosis is normal cell division, but sometimes that can cause cancer. Cancer is the uncontrollable growth of cells. So you see when you have normal cell division, you're dividing into two, and then this new cell also divides into two, but sometimes you get a damaged cell. And when you get a damaged cell, then that damaged cell will hopefully get found by the checkpoints in interphase and will go through what's called apoptosis. And apoptosis is programmed cell death. It recognizes that it's not a good cell and it will die. But sometimes it doesn't get caught. And when that damaged cell doesn't get caught, then it can go through its own cell division. And there's really no stopping it because it will just divide uncontrollably, very quickly, continue, until you are forming a tumor. And tumors are abnormal groups of cells that develop when you have uncontrolled cell division. There are two types, benign and malignant. Benign don't invade nearby tissues. Malignant are cancerous tumors that can move elsewhere in the body. There's a number of things that can lead to cancer. One of the big ones is smoking. There is a positive correlation between cigarette smoking and death due to cancer, right? We know that chemicals in tobacco are carcinogenic. And so some key terms that you should just be aware of when discussing cancer, there's oncogenesis, which is the formation of tumors, oncogenes, which control the cell cycle and cell division. If mutated, those oncogenes, they can cause cancer. And then metastasis, which is the movement of cancerous cells from the primary location to a new location. So this would be like if you had breast cancer and it moved into your lymph nodes, and then you have a different type of cancer from there, but it all started with the original breast cancer. Okay, so we're going to move into the stages of mitosis now, but before we do that, you need, again, some working definitions so that you are clear when I'm saying these terms, what they mean. So there are forms of DNA. Chromatin is this like squiggly bowl of spaghetti. It's a mass of DNA. It's present during interphase, and it will then condense into chromosomes during mitosis. So during prophase, the first part of mitosis. So chromatin is squiggly, long and thin DNA, and then chromosomes are these like X-shaped things. They are coiled DNA, so they contain all of your genes, but chromosomes themselves are coiled, supercoiled DNA, and that's how you actually go through mitosis, is by forming chromosomes. And then within a chromosome, there are terms that you should know as well. So each chromosome looks like an X. There are two sort of sides to a chromosome, and those are called chromatids. Chromatids is a half of a replicated chromosome. So one side of this chromosome is a chromatid. They are identical to each other, each side, so they are called sister chromatids. Within a chromatid, you have a gene. A gene is just a segment of DNA that's going to code for a trait, whether it's hair color, eye color, can lead to height, it can lead to anything, right? Any gene is going to be found on a chromatid in the same spot on both sister chromatids. And then the centromere is what holds the two chromatids together. So this allows the chromatids to form into the like typical X shape of a chromosome. Continuing on with structures that you'll need to know for this, there's a centrosome. I know, there's so many terms, I'm sorry. There's a centrosome, which is the microtubule organizing center. You have two centrosomes here, okay? On each side of the cell, you'll have two centrosomes. From those centrosomes, you have centrioles, which form spindle fibers during mitosis. So these like Spider-Man like web shooter things, those are coming from a centriole. The spindle fibers are the microtubules that come out of the centrioles, and they connect to the chromosomes and move the chromosomes around. This is how chromosomes move within a cell. And then the kinetochore are structures at the centromere where microtubule spindle fibers attach. So remember that the centromere is this piece in the middle that connects the two chromatids, and on that there are kinetochores, and those kinetochores connect to the spindle fibers, and the spindle fibers are being shot out from the centrioles. Okay, I know, tons of vocab, I'm sorry. So let's talk about the stages of mitosis. There are 4. Prophase, this is where you're sort of preparing to go through mitosis. Chromosomes thicken, they're condensing, you're starting to actually see chromosomes and not chromatin anymore. Sister chromatids are joined by centromeres. Centrosomes move to opposite poles, right? You see these little like macaroni and cheese noodles, right, moving down to opposite sides of the cell. The nuclear membrane disappears. That's what these white lines are. If the chromosomes can't move if they're stuck inside the nucleus, so the nuclear membrane has to dissolve, and those spindle fibers form from centrioles and attach to the kinetochore at the centromere. Okay, so these spindle fibers are starting to shoot out and actually attach to the chromosomes. This will allow movement through the rest of mitosis. In the next phase, metaphase, it just means in the middle. This is super straightforward. Those spindle fibers connect to the centromeres, and they just line up the chromosomes along the middle of the cell. That's the whole thing. They just line up in the middle. Our next stage is anaphase. This is when we're pulling the chromosomes apart. So now our spindle fibers are starting to contract. They're going to move back towards the poles, back to the centrosomes. And then once they are separated, the chromatids are now separated and in a single chromatid. They're no longer sister chromatids together forming one chromosome. They've split. Once they've split, now each of those chromatids is considered a chromosome in its own right. So they were chromatids, but now during this phase, we're going to start calling them chromosomes. Yep, I know. And then finally, telophase happens. Telophase is basically the opposite of prophase. So we have our spindle fibers start to dissolve, go back where they came from. The chromosomes start to decondense and are no longer visible because they are turning back into chromatin. The nuclear membrane is starting to reform, and cytokinesis will then occur kind of concurrently as telophase is happening. It will occur at the same time, the splitting of the cytoplasm, but for the purposes of this slide, we will only really be talking about what happens during telophase. And so, again, we're just doing the reverse of prophase, and we are forming 2 identical nuclei now. Our final step is cytokinesis, which is just the division of cytoplasm and organelles. We have officially formed 2 distinct cells with their own identical nuclei, but they are 2 new cells. Animal cells and plant cells do this differently. In animal cells, there is no cell wall, so what's called a cleavage furrow forms, and basically it's just like pinching in between the 2 nuclei and pinching enough until the cytoplasm splits and you have your 2 cells. But in plant cells, you have a cell wall, so a cell plate forms, and it's a much more distinct and hardened way to divide the cytoplasm because of that cell wall. And then finally, mitotic index. Yeah, because of course we got to bring math into this. So the mitotic index is just a formula to determine how many cells are actually growing and actually going through mitosis when looking at a slide. And so what you do is you add up all the cells that are in or going through the different stages of mitosis, prophase, metaphase, anaphase, and telophase, divided by the total number of cells that you see, and multiply that by 100. This is really important in determining which tissues are growing. It's a great way to predict the response of cancer cells to chemotherapy. If you are going through chemotherapy and you have still a really high mitotic index, that means that the cells are not responding to the chemotherapy, meaning that they're still replicating at quite a high number, even though chemotherapy should be preventing that growth from happening, ideally. So Really important. We will practice this, don't you worry. But great job. Proud of you, and I'll see you in class.